collect.
No, I've seen it. That's what I started doing in the end, and then I closed the rest of my brain. I think I kind of preferred my method because then I had them in a space where I could easily see them, and then it was quicker to put them into each page region.
But anyway, there are several containers. You can pick out from one container at a time. Next time from next container. So you have more. Yeah.
Professor, I have a question regarding the collecting the insects for pest management, for urban pest.
Yes.
Because Nico took you like last semester, right?
Yes.
They said that you needed it on the trap.
I'll give you a trap. I'll give you a trap. But you can
But
also
I
collect.
think you told me like yesterday or something.
You can collect in hand, in faucet.
By hand?
Yeah, you can.
Because like I just wanted to confirm that. Like,
Yeah, you can. No problem.
so I can just take a picture of it, like peel
Yes.
back bark, see termites, take a picture, and that's it.
Right.
That works.
Yes. I will announce that later.
Okay.
Yeah, I'm very. I remember it was very difficult to get things on the trap from where I was.
Yeah.
There was nothing that was coming up.
Yeah, you have to put a probably outside or in dirty place.
Can we try use? Are we allowed to use traps that we like learned in different classes, like the dish one with the soap?
Yeah, pitfall trap is fine. You can use pitfall trap. Yeah, okay, let's start.
With the cup.
It's already 11:04 or 5. Okay, we'll start.
Oh wait, no, because those don't attract pests all the time. That's why.
Okay. Good morning, everyone. Today we are delighted to have our speaker, Zhang from Zhejiang Agriculture and Forestry University. Actually, I know him for more than 10 years. He got his PhD degree in Zhejiang University. Then shortly after, he went to University of Guelph in Canada to do postdoc research work. After that, he went to Indiana University in Bloomington, Indiana, for a research associate position for a few years. Then from 2013, he went back to China. Now, so where he studied urban entomology, insect genomics, etc. So for about 11 years. His expertise in insect behavior, physiology, toxicology, and genomics. He's especially interested in the study of termites, fire ants, and soldier flies, black soldier flies. And he and his colleagues developed a termite monitoring technology, which is, I think, currently the most accurate method for detecting termite activities. He published nearly 100 scientific articles and also had 5 patents. So welcome.
Okay. Good morning. Thank you, Chang. Thank you for. It's my great honor to come here to give this talk. Actually, I'd like to share some works conducted in my lab. The first things I would like to share is how does oleic acid trigger burial behaviors in termites. So actually, before that, I'd
Could you speak up a little bit? There's
speak
you
here.
have to speak up.
It's louder.
It's just a long room.
Oh, okay.
Yeah.
Yeah. So now this one is okay. Sure. Yeah. So before that, I last night I talked with Chang. Chang said you probably can talk about the equipment, the device, which is used for the remote monitoring termite activity. So now today's topic, I will divide it into 2 parts. One is short part is to introduce the termite device detecting termite activity, and another one is talk about the oleic acid trigger burial behaviors in termites. Oh, okay. Before that, so I'd like to give some very short introduction. It's for my research projects ongoing in my lab. So one's on the termites' social behavior and associated mechanism. for instance, such as burial behavior and tetraphylaxis. So I will give a talk on the burial behavior today. And another topic will be on bio-termite side. We specialists use the plant extracts, for instance, for the essential oils. So we use the nanoparticle to package it as essential to improve the efficiency for the pest control. And another project is on the ongoing on termites, is on the wireless systems. Is for monitoring termite activity using electromagnetic induction with non-looping method. So this one is developed a few years ago. It's in 2018. Now after a few years developed, now this method is this device. We can see it's the highest detected rate in the world, we can say. Okay, anyway, so I will give introductions for this one. So it's for the termites control, we all know. So the very common method used is for the termite side, is to confirm a barrier treatment. Put the termite sites in the soil to form a barrier treatment. And now many alternative approaches, such as base stations, have been widely used. It's for integrated termite management. So we can see the lots of this kind of things as is market. So when we use this all kinds of is a manual device. So what's the shortcomings of this manual device? Is sometimes we don't know if there are any termites inside of the bait. And when you treat it, you have to open it. It will disrupt the termite activity. Termite runs away. So it will affect the control efficiency. And so it's highly demanded to develop a remote monitoring device. So when we can sit in the office, you can know this station, there's any termites, not, yes or not. So actually, there is a few things we have to think about. It's for the remote device. One is accuracy. So accuracy is very important, right? So if the base stations give the signal, if it tell you is there is termite. So when you open it, there is no termites. How is it coming? So it's should be a very high accuracy, especially it's in a little bit long time. For example, it's more than 1 year or even 2 years. So one is accuracy. Another thing is durability. So this device can be used for a little bit of long time. It's not just used for a very short time. 1 month, 2 months, it's not durable. It should be used for 1 year, probably, or maybe 2 years, it should be perfect. And another thing should be the real time. So when the termites consume the bait, so it can tell you immediately what's happening in the base station, if there is any termites there. So actually, to get this accuracy, durability, or real time, there is not so easy. So because it's if we use the electrical products to give the signal, usually electrical products is always underground. So it will affect the electronic accuracy. And also, how can get the information? How can trigger the information? How can get the information from the underground to your office? So there's a few products already. It's the first things I can see the published papers is actually in the States. Is in the Sunayao is in the University of Florida. So he developed ESP products. Actually, is sold to the Dow Agro, I think. And after that, there is a few a few methods use it. Use this wire, use this metal springs, use the metal particle, or use the graphite, or use the metal powder, or use the infrared looping technology. All can see this kind of things. You can see it. But the problem is for all kind of this kind of method use it, this kind of device, they show the very high false positive rate, and also the time is very short. I mean the durable is just a short time, short life. So how to improve the accuracy, how to improve the real-time warning, and how to improve the durability. So this is the hypothesis. We use it a few years ago. So we work with I work with companies, the companies in China. So we hypothesis. We think we can use one code is non-looping. What means it's termites consume the wood. So we treat it as a termites wood states are no longer as the electrical circuit part. Instead, the wood and all the particles are just used as supporting material. So this thing is a bait separated from the electrical circuit part. That's one we call it non-loop. Another thing is we use the electromagnetic induction. So this is for the signal transduction. So we combine this together. It's called Deacon electromagnetic induction with non-looping technology. I give the short name is called DEMIO. Anyway, so I already published this paper in Applied Science is recently. So use this technology to detect the presence of the termites, which is show the changes in magnetic amount during termite consumption. So how does it work? So when the termites consume the wood, there is some glass particle already installed in the wood. So when the termites consume the wood, so the glass particle will move out. Above that, there is a magnetic bar. So we can see the magnetic bars drop. It's very clear. It's caused by the gravity. So it's very accurate. So when the termites consume the wood, the glass particle drop, and then the magnetic bar drops. So there is a detector. So the electrical detector here, they can tell the magnetic amount changes. So if the magnetic bar drops, they will tell you there are some signals here. So this is our principle. It's when you tell it, it's quite simple, right? But it's before that, we spend a long time actually. It's try to figure out how to do these kind of things. So
it's also water resistant, right? Because water does not.
Yeah. So the first thing we did not put the water resistance,
Right.
so it affected by the water.
Yeah.
But after that, we can put these things is use the seal it.
Right.
It can reach the IP68 level. So it's water resistance. So now we already developed two devices, actually two systems. One is called as a semi-automatic device. This is information bar. It's a kind called as a bait. Another one is called the signal receive. This is background is termite management. It's just a computer or some software. So this one is look like a signal, look like a cell phone. So you use it just to go into get close to the station. So the signal will tell you if there is any termites or not. So this we call it. It's called semi-automatic. It's not fully automatically. Another system is called fully automatically. So we use the LoRa technology is for the communication. So there is a device here, and the device gets the signal is to the concentration is a gateway. When the gateway gives the signal is to the network server. Actually, this is a very signal is wireless. So when they get a signal, you can get the signal from the computer, from the see the background. So this is also very fully automatic system. So we have two systems. One is semi-auto, one is a fully automatic system. So now take a look at the applications. So this is not serial. This is already applied. Since 2018, we have 4, 5, 6 years already use it. So even in the very famous place, is a museum in China, is the Forbidden City. So recently we applied this one. Actually, in Beijing, there's not so many termites, but they worry about if there any termites comes. So say that we don't care about how much it costs, but we care the historic stuff not be damaged. So just put your stuff here. So this is a Forbidden City. And also we use some dams, some traffic as and most of it for the houses. So I'll give some examples. One is in the Zhejiang in China. In Zhejiang is a lake, is a reservoir. So this one is called the Siming Lake Reservoir. It's a very big reservoir. So the water is used for the drinking purpose. It's a very big one, and we put the as I remember, I did not say how many. It's about 300 stations over there. So after 1 year and after 2 years, we still can see about 95% accurate rate. 95%. So this one is in Hangzhou in China. It's called the Xilin CO Engraving Society. So in this place, we can see it's more than 99% accurate rate. And another thing is in Guangzhou, in southern city in China. In this place, as after 1 year, we can see that it's 95% accurate rate. So in China now this systems already applied is about nearly 200 sites. So one site means I just now mentioned. So the Forbidden City is one site. So the China Academy is one site. So there's almost about 200 field tests. Now we are thinking about the international application. So maybe in the near future, we can see these products in the States. We talk about this kind of things in Australia. So in March, I will go to Australia to talk about this kind of stuff. So these products we show the accuracy, durability, and real time. So now it's very practical to be used. It's for the remote control termite activity. So for this topic, I just give very briefly talk about the principle, how does this device works, and give some application to tell you this is a wide range used, it's high accuracy and real-time warning system. So far, I think it's I cannot say it's number one, but actually it's number one. Now after that, there are lots of companies just follow our step to do some kind of stuff. But this kind of things already protected by some patents. So it's not so easy to do us. But they can use different principles to develop it. So now in China we have a few companies to work on this kind of devices. So this is the first part I already talked about. So when after the seminars, if you have interest, we can talk about more. Okay, so now we go to the second part. Is Actually, this is similar as I give the secret in the department. So this one is how does oleic acid trigger beauty behavior in termites? This one is more like the basic research. Didn't ask me, so why you work on this kind of things? I said it's curious. But anyway, so it's a basic research. So human has a curious, right? So we want to dig out. So why? So now I tell why. So it's the answer is here. It's oleic acid triggers behavioral behaviors in a termite population through the olfactory sensory system. So how I tell you the story for this one. So we all know the social insects is living in a very crowded population. So it's very easy to get the individual very easy to get sick. So it's caused by lots of factors. So one factor is caused by the death body, right? It's a death individual. So in the population, there is some death individual. So usually it will cause some bacteria, some fungi, or some infected diseases. So the colonies have to treat it. Look at it's a human, right? So we do the same things, right? It's when the people with old or died or something else, we have to bury it, we have to burn it, we have to treat it, right? So different ways. So this is in order to keep the population healthy. Good thing is we also see this kind of phenomena in social insects, in bees, in ants, in termites. But they play different stories. Bees can remove the infected fluid from the cells. They can just move out. And ants can transform or bring the dead individuals to a designed area. But it's for the termites. It's a flesh body, they probably can eat it, but it's for the a little bit older dead body corpus. They just bury it. So cover the dead individuals with soil or with dust. They just bury it. So bury behavior is only found in termites. I mean in the insects, only found in termites. It's not other insects. It's very interesting. So why cause it? But I don't know. So why cause it? Why cause it? The termites can bury it, but any insects cannot bury it. So I don't know why. So it's a curious story. If we have questions, if we have this kind of things, we can dig it out. But termites has this kind of special features. They can bury the dead individuals, and which is different from the ants or the bees. So here's talk about the one termite samples used in my lab. We use the Formosa termites, which is also the main termite species in the States, especially in the southern states, in the United States of America. So the termites, for the Formosa termites, we put the death individual just in the petrol, as put on the papers. This is the death individual. So after a few hours, as 2 hours, we can see the termites start to bury it. After 4 hours, it's almost buried, and 6 hours is almost completely buried. So it's very clear. So just take a few hours, they can start to bury it. So now the question comes: how can cause it? So I mean, which kind of signal tells the living termites to bury the dead individual? So we think about it could be the accumulation of chemical compounds in the corpus may cause this kind of behavior. It's very normal thinking, right? It's a signal should be from the dead bodies. So what kind of signal it is? This is what we want to know. So the first question, so what are the chemicals in the corpus of famous termites can trigger the burial behaviors? And are the fatty acids, I mean here is a polysemitic acid, stearic acid, or oleic acid. This kind of fatty acid can cause these termites burial behavior. Why we think about the termite fatty acid? Because it's when we check the termites corpus, when the termites died, they can release some chemicals, some signals. For example, phenol, idol, or octone, and also lots of fatty acid. So we check it if there are any, which kind of fatty acid it can be released. Actually, so we can see the palmitic acid, we can see the stearic acid, but the most part are oleic acid. So now the very common questions, right? It's all this kind of oleic acid, we can see lots of signals comes. So this chemical can be caused the burial behavior. It's very normal considered. So we check it. We use the polyic acid. We use the stearic acid. We use the oleic acid. So when we put this acid on the field paper, and then put on the sand, on the sands. But it's for the palmitic acid. There's no any signal. Just blank papers. It's no any sand found. It's for the stearic acid. It's no burial behavior. But it's for the oleic acid. See, it's almost about 2 hours. They start to bury. But after 4 hours, almost buried, and 6 hours, completely buried. That's very clear. It's very interesting. So now the conclusions, first one. So the oleic acid released is from the corpus of Formosa termites can trigger the burial behavior. It's in the living insect in termites. So it's very clear. And also oleic acid no need to combine other signals. So only oleic acid can do it. So this chemical can do it solely. So now goes to the second question. So this oleic acid can cause a burial acid. So what's the mechanisms under that? So it's easily to think about it. So it's oleic acid is a signal in the air. So the living termites probably can smell it and then cause this behavior. So we need to approve it. It looks reasonable, it looks logical, but we need to prove it. And how to prove it is one thing is to see if there are any odorant body protein is plays any roles, and which one is will be played. So now take a look at the olfactory sensory. So basically, it's now it's already very clear. So it's for the first step is interest of odors into antenna. So when the odor molecules enter into antenna and bind to the odorant body proteins, it's the first step. And then OBP protein, OBP, transform this molecule into the receptor. It's called the odorant signal receptor. Transfer over there, and then cause the olfactory sensitive nervous. Then cause the burial behavior. So this is olfactory system works. So now we need to approve it. It works on the oleic acid. So now first things we try to look out which odorant body protein it works on this system. So we did a lot of transcriptional analysis. you kept the different tissues and doing a very big transcriptional analysis. And this thing is just tell you it's the antenna expression, so the gene expression antenna compared to the other bodies. So we can find as different OBP. Most of them all high expressed in the antenna. So OBP is mainly expressed in antenna. This is the FPKM's data. It shows some OBPs. We did not get so many OBPs, but we get about 10 OBPs. It's not too many, but we only can find 10 about it. So it's one is OBP7 is highly expressed, especially highly expressed in the antenna. So now we have 10 OBP, but we'd like to know which one it is, right? Which one it is played in this processing. And the first steps we're using molecular docking. So this one is quite simple actually. It's not no need to do the experiment. We just use the docking. We found that this kind of OBP. Only one OBP, OBP7, which is highly expressed and also has a very high binding affinity. So this tells us we can use this one to check if this one works in the system. So now we take a look at this OBP7. So this is qPCR data is from the different parts of soldiers or the works. So we can get the conclusions look like this. OBP7 was highly expressed in antenna. OBP antenna is the major organ sensing odorant molecules. In order to approve the OBP is working in this system, we need to do some in vitro expression experiments. So we get the OBP genes, clone it, and get into the expression vector, and transform it, and purify, and get the protein, and purify. We get the purified overexpression the OBP7, and we cut this purified this protein, and doing the fluorescence competitive binding assay. So we try to see this OBP can bind to the oleic acid. So this is a very typical step to prove OBP works. So this is the background actually. OBPs work with the NPN. They have shown the fluorescence. If add some signals, for example, here is some polysmatic acid, stearic acid, or oleic acid. They can bind with NPN, or they compete with the NPN, and to show the reference signals. So especially for the oleic acid, is here. This one is strongly competed with the NPN. So which means oleic acid can strongly bind to the OBP7. So the fluorescence intensity of OBP7 decreased significantly after binding with oleic acid. So this data is not enough to prove the OBP7 is caused by this kind of behavior. We also use the RNI treatment. We'd like to use the we'd like to use the genetic modified population, but unfortunately in the termites, it's very difficult to get it. So we have to use the very typical, very I mean the low levels. Now we can say that we can use iron. We have to use the RNI stuff. So its iron efficiency is about 70 or 80% efficiency. So when we use the palmitic acid or use the oleic acid or use the stearic acid, they show the different EAG response. So it's for the oleic acid. So when the termites treated by the double strength OVP7, which is INI treated, so the signal is significantly dropped. So which means it's oleic acid can just cause these things. But it's not for the palmitic acid or for the stearic acid. So this one is confirm the previous experiment. So the conclusion is after OVP7's influence, the antennal signal sensitivity to oleic acid decreased significantly. So OVP7's antennas plays important roles in the positions of oleic acid. Now we can give this conclusion. But we can give a little bit further. So we can see the behavior, what's the behavior causing. So when we OVP7 RNAi treatment, we take a look. For our treatment, it's for the control. Its control is for the GFP RNAi treatment. This is RNAi is OVP7. So when we can see the after GFP RNAi, just there is no any dust. So it works. It proves that OVP7 is plays roles in this bearing behavior. So we can say OVP7 is an important odorant binding protein for the bearing behavior. So we just send these conclusions in the insect biochemistry and molecular biologists. So the conclusion here is that oleic acid triggers bearing behavior through an odorant binding protein. It's very clear. And also proves that the olfactory sense system is associated with the oleic acid triggered by the bearing behavior in termite populations. So it proves that with hypothesis, so the olfactory sense system works in this behavior. So now we go a little bit further. So the question 3. So what's the core of this system? Because it's in the olfactory system, the core of olfactory system is a receptor. It's called the odorant receptor. So we try to figure out where is this receptor. If we can find this receptor, it can be complete. It will be very perfect to explain this behavior, and also it will be a significant contribution to the basic research, right? So what's this one? What's odorant receptor in olfactory system? Actually, this system is oleic acid receptor. In the body, there is oleic acid receptor. It's called FAR4. This kind of stuff. It's used for the lipid droplet. It's in the cells, but definitely here is totally different. It's not this kind of receptor. This kind of receptor is here. Receptor is the odorant receptor, because oleic acid is look like acting as a phenomenon. So it will be a totally different receptor. It's not like a common we take as a receptor. So this receptor is to bind the phenomenon. So it will be an odorant receptor. So we try to figure out where's and what's this one. We're doing tons of work to try to. The first step, we to get how many odorant receptors in the termites, in the Formosa termites. We're doing lots of jobs for the transcriptional analysis, for the for the genome reannotation. So we use the genome sequence data. It's already published, but there is some data missing. So actually, the quality is not so good. So we also did a lot of transcriptomal analysis, different tissues, antenna, heads, body parts, legs, possums, and combined with the genome. So we finally get some odorant receptors. We found 57 Formosa insects, Formosa termites ORs. So 57 is here, 57. And this one is showed some ORs has a little bit higher expressions during at least one tissue. And this one is showed some ORs is highly expressed in the antenna, but this is all just to, I mean, it's not so significant. But the only thing here, use this approach, at least we get the 57 ORs. So now we need to think about which one is the core, or maybe the 2, or maybe 3. So at least we need to find one. 57 is still a big, right? It's when you do it, it will be cost lots of times. So we try to doing some bioinformatics analysis. We use the mechanisms, likely trees construction. We see the sequence correctly. So the first thing we take a look at the numbers of the exon. And another thing is we take a look at the DN solution of DN to DS ratio. So this is mainly associated with social activity. We can see the published papers in the B's, is ants. So we choose the numbers of exon or the ratios of D and DS. So it's for the gratitude OS, we can see different as a family. We can see the different family is family 1, 2, 3, 4, 5 until 5. But here, you can see 1 and 3. It's highly associated with the high numbers of exon and high numbers of ratios of DN to DS. So it's probably indicated that family 1 or family 3 they may be associated with social insects, social behaviors. So at this place, for family 1 and family 7, we still have 36 genes, still lots. But anyways, at this step, we cannot figure out which one is the easiest one. So we did it every OS. We doing the I&I, and to see what's the behavior, what's the value behavior happens. So we doing the every genes silence 1 by 1, 36 genes, and good things. Luckily, actually, I can say we found one. It's called ORX. Actually, it's maybe it's OR. We have not given the name yet, so we just give the ORX. So this just means only one OS is practice associated with the berry behavior. So this is the I&I approach. So we take a look at this genes. We spend lots of time and finally to get one. So we take a look a little bit deeply to see the ORX I&I. So we can see that after I&I, there is no dust count, but it's no I&I treatment. There's lots of dust covered with the dead body. That's the one thing. Another thing is we'd like to take a look at the electrostatic activity. So we use the EAG. It's called this electro antenna graphic. So we take a look at what's happening after after RNI treatment. So definitely yes. So after RNA treatment, we can see the signals here. It's for the control. As no RNA treatment, the signal is pretty high, but after the treatment, the signal is pretty low. It's very significantly. The only happened is to the oleic acid, not happened to the other fatty acid. So this is the RNI treatment. And one more step, we have to approve that the ORX is the core. So the core has a high affinity, which means it can bind to the oleic acid to trigger this behavior. So we take a look at some data is for the marking date. Yes, it shows that ORX has a high affinity with oleic acid. Especially the oleic, we use the 170 different phenomena, different chemicals to treat it. Above the fatty acid, oleic acid is showing a very high affinity. So now the last step, as we'd like to see if the ORX is in vitro expressed in exogenous octosis. So this one can change the ion currents after putting into the oleic acid. So we express this gene in the exogenous octosis and put the oleic acid and other chemicals to see if there are any ion current changes. Yes, we see. As it's for the oleic acid, we clearly can see the ion current change. So based on the RNI treatment, based on the binding experiment, binding on the marking, now we can give the conclusion. CFORX can bind to the oleic acid, act as oleic receptor to trigger the building behavior. So lately we will give the oleic acid as a formula name, and we will give maybe 1, 2, 3, 4, anyone. So just make sure, and then we will prepare this manuscript. So the OBPs we already submitted and now in under review. But ORs we think about to give a little bit a good general. So this is what we are doing here. So now give the summary. So here I tell the story of the oleic acid to trigger the bearing behavior. So the first step is the signal as the oleic acid is released is from the corpus, then detected by the living termites, and then the oleic acid enter into the antenna and come to bind to the OBP, odorant binding protein, and odorant binding protein bind it and transfer this chemical, transfer the oleic acid to the receptor, and then trigger the olfactory sensory neuron to trigger the berry behavior. So this is finally we give the conclusion. So at the last, I'd like to give thanks to actually this work is already done in my lab for a few years. It's already five years. It's a long time. As I'd like to think about the Hongyue Li is here, and Liu Jiahai is here, and Zhao Weishun is here, and Bosen Cheng is here. So these four persons gives a very significant contribution to this work, and also give the thanks to my colleagues in my group. So my group is in Zhejiang Agriculture and Forestry University. It's called Insect Physiology and Toxicology Team, because we conduct the most workers on termites. So people also call us as a termite group. Anyway, and also the funding grants, the federal and the local province the grants, and also from the company, and also from my university. So, okay, I think this is my talk, and thank you very much. now take questions if you're interested.
Hi, interesting talk. So I have one question because you basically proved that oleic acid, the termites responds specifically to oleic acid and not to other fatty acids, right?
Yeah.
And I was wondering, because oleic acid has a trans isomer called L-acetic acid that is not present in insects. And I was wondering if you were also thinking on trying that trans isomer to see if something that insects don't produce or are accustomed to respond similar to oleic acid or not.
You mean the oleic acid where from?
L-acetic acid, the trans version of oleic acid.
I didn't get you saying. Say
The
again.
trans isomer of oleic acid,
Oleic acid.
which is called L-acetic acid.
Trans isomer. It's kind of a trans isomer.
Well,
No, I didn't get you. I'm sorry. So what's the
so
question?
it's basically the same, just the double bound bends the molecule in a different direction.
I think so. Yeah.
Okay, so but the L-acetic acid is not present in arthropods as far as I know. And I was wondering whether they would recognize it similar to oleic acid or not, and if you have thought of testing that because it's very similar.
Does it occur in nature?
Yeah,
It just doesn't occur in insects?
that's not very insect.
That's really cool.
We did not test it. The probably is yes. You think so. But here we are thinking about the oleic acid is as just can thoroughly simply can work it.
Right.
Okay, clearly. So
If you put oleic acid on a living termite, will it bury itself?
good question. Good question. So we tried. I asked my student to try this one. Yes, they fine. They will fine.
They will fight?
They'll fight
Yeah.
back, yeah.
Yeah, they will fine.
So it will not bury itself.
So the living insects is fight with the living insects with the oleic acid.
But I mean, the point is that the termite itself is trying to bury itself, is what you're asking.
No, that's what he was
No, I know, but you're saying, would the termite itself agree? Because it's like, yeah, this needs to be buried.
How long would termite dead for? When you with the very first experiment, when you put the dead termites, how long were they dead? Like how were they killed?
No, yeah, this is we use different ways. Here we use the code. We code immediately just code it so the data is put it here. So put it here. Basically, it's for the very flesh that is, the termites can eat. If the termites are hungry, they can eat it. But after a few hours, termites just bury it. So here the put it here is a flesh treated corpse.
So is there a variance in how long you hold the corpse? Like you freeze kill it.
Yeah, freeze kill.
Within 5 minutes, 10 minutes, you transfer it.
Just
to
1 to 2 minutes. Just very.
Have you tried like how long? Like hold it for 2 hours, 4 hours?
Right. How long,
Yeah.
how long does it take
Before
if
you
it's like
introduce.
not worth eating? Right, is that what you're asking?
Yeah, I'm wondering if
When does
how,
it shift?
Right.
So you mean the termites will come will
No,
be
how long
recovered?
would you hold a dead termite before you introduce it into the live ones? Like, is there a variance there? And the other is, like, if they were killed in a different way, would they have different reactions? Like pathogen killed, parasite killed, or dying of aging?
Yeah, this is a good question. Actually, so we use this a few hours to kill it. And so one is heat, one is cold, one is just use a needle to, to kill it. But they look like a very similar, very similar thing. But we have not tried to use the pesticide. Maybe it will tell a little bit different story. But basically, it should be the same, I think. Very similar. Yeah.
First of all, I thought that was fascinating. That you went from the beginning all the way to identifying the gene. But I have a question about the first part of your talk, about the sensors detecting
Yeah.
termites. Because I assume that what was in the, and maybe I missed some of it in the beginning, but that contraption has wood in there, right? So the termites are eating the
Yeah.
wood and that makes the magnets fall.
Yeah.
I was curious about what kind of wood, and would the kind of wood you use affect the time it takes for them for the response? So if you have a
Yeah,
hard wood
it's a-
versus a soft wood?
It's actually the termites. It's a wood. Different wood has different feeding activities for the termites. We use a pine wood. It's a kind of soft wood.
Right.
So the termites.
So that
different
would
kinds of
determine how
different.
fast you get the response. If the termite is eating, say, mahogany in an old temple, would they be, better at, I mean, is there a time thing? How much did you try to sort of standardize how fast you get a response from the termites when you put the instrument in there?
It depends actually. It's for the different of kinds of termites. It's for the older termites. It's very quick. If there is termites around, is nearby, it just costs them a few weeks, 2 weeks, 3 weeks. And for most termites, it's very quick. It's a few weeks. But for some other termites, it's here happens in the New Jersey or in the Boston area. This kind of termites will cost a long time.
Is that a problem? I mean, weeks
No,
sounds like a
it's-
long time. I want to know
it's-
now. Is it termites?
No,
yes or no?
it's just a feeding activity. So it's different kinds of termites have different kinds of feeding consumer. And for the termites common with here, their feeding activity is not so popular.
Okay.
So powerful, not popular, so powerful. Not like for most termites or not like the others termites.
Thank you.
So does the oleic acid work as an attractant? Because they go to bury the dead termite. So
Good
technically,
question.
couldn't you
Good
use
question.
it as bait? Oleic acid?
No, I don't
No?
think so. I don't think so. As termites, you probably can try that. I not completely say no, because these things is just tell the termites try to bury it. It's not to tell to feed it, to try to feed it. So how can you use it? It's for
No,
base stuff.
because don't they pick it? They pick it up olfactory, right? So does that mean they go to it to bury it?
Mm-hmm.
So that means that they are attracted to it, correct?
Yeah, but it doesn't sound like that. It also it doesn't seem like a large proportion of them. I mean, it takes 6 hours for a few grains of thing. I mean, it's not like everybody just comes running. That's what it's.
Yeah, maybe there's some other factor to check it.
Right.
But the only we can tell here is oleic acid can cause this kind of
Right.
behavior.
Got
Yeah,
it.
so
It's a great system to work. I mean, this was clearly a labor of love. It was like, oh, I wonder about this, and then take it all the way to figure it out. I mean, you saw how quantity of different things that had to be done to connect a behavior to the actual source of that
markers, yeah.
behavior. That's
Yeah,
really
yeah.
cool.
yeah,
That's,
yeah. Really.
Hi, thank you for your talk. It was really interesting. I was curious, how far does a live termite need to be in proximity to the dead termite to pick up the signal?
Yeah, very good questions. Actually, so we did not test it how far it is. We only put in the petri dish, and they can test it. So it's just 10 centimeters or 20 centimeters, something like that. We did not test the very, very far distance.
Do different casts release different amount of oleic acid when they die?
Yes, I think so. So the only test here, so we can see this kind of behavior is work. It's not for the soldiers.
Yeah, that was
Soldiers,
interesting.
there's no any behavior.
Just the
Soldier
workers.
only fight, only works.
So soldiers do not have oleic
Yeah,
acid.
no, they don't respond to the oleic acid. There's no response at all.
Oh no! I messed up. I
Yeah.
messed up what I heard then.
Yeah.
I thought I thought it was said that the soldiers don't have oleic acid, so they don't bury the soldiers.
No, I don't know.
No.
Not that
So-
the soldiers
Right.
don't react
Yeah.
to it.
Soldier is not reacting to oleic acid, definitely yes, and the soldiers also not reacting to some other stuff. It's here we can see this kind of things only in the work.
Those very good.
Maybe one more question? Okay,
Oh.
last question.
So now when this oleic acid triggers the response, is there an application to it? Is there something you can do with this knowledge now, or is it more of just a fun fact?
Can you say again? Yeah.
This is basic science,
Can you
people.
do anything? Can you do anything?
有没有应用前景？
Any application?
You mean the oleic acid can be used apply for the pest control something like this?
Well, just like if you can do anything with this knowledge for termites, like treating them or working with them, or this is cool, this is how it
It
works.
wasn't just,
Yeah, that's a good question. So we are thinking about it. We are not so sure. So how can you use it for especially for the oleic acid? Probably yes. I think so. Probably yes. Because it's just not it's sort of it's put the oleic acid on the living termites. They probably can fight, so it will damage the colony.
Check it. Are there any questions on the Zoom? I don't know.
oh, right. there's people
All right,
online.
there was
Go
a
to Zoom.
thing.
Go to the chat. Go to the chat. Any questions from
What's
the
that?
audience in the Zoom? Is it unmuted or muted? I don't know.
I think we can see. Okay.
Check it.
Oh, see you later. What a nice talking to you.
Thank you.
Really interesting.
Chat.
Because you're staying, you're taking the class?
Yeah, I was gonna say.
Where's the chat box?
I don't
Any
know.
questions from the audience here? I cannot see. Participants only 3. Okay. That's all. Don't worry.
Okay.
All right. Thank you, everybody. Okay. That's fine. All right. We'll stop here. I don't see any. That's okay. All right. That's good. Very good talk.
Is there any other questions or?
No questions.
I just kind of building